All right, hello everybody. This is my recording where I'm going to attempt to talk to you guys about cellular respiration. This is the process in which the cell will transform or convert the potential energy in glucose, in the structure of glucose, over to potential energy of ATP. Of course, there are going to be many reactions in between this. We're going to see a series of redox reactions. Those are the oxidation reduction reactions that will initially work to kind of break the bonds of glucose. Glucose gets oxidized to CO2 and water. In the process, NAD will become reduced to NADH, and FAD will be reduced to FADH. And then those reduced NADs, well, NADHs and FADHs will work in the electron transport chain then to produce ATP. All right, so that's sort of the overall gist and where we are going with this. As far as cell respiration goes, this is found in all organisms. That's pretty neat, I think. Plants, animals, eukaryotes, prokaryotes, they all use cellular respiration to break down glucose to CO2 and water. But this pathway is also flexible in that it's not just for breakdown of glucose to CO2 and water. You could instead use the pathway to build molecules like fats, other carbohydrates. Maybe you ingest glucose, but your body needs some other sugar. You can convert it. Also other compounds, things like lipids or possibly amino acids, things like that. We're going to focus, of course, on how glucose is used to make ATP through cell respiration or fermentation. But I do want us to look at this process too. In addition to the pathway being flexible, I really want to point out to you all that cellular respiration and photosynthesis really support one another, and that ultimately all glucose comes from those organisms that can carry out photosynthesis, things like plants or algae. They are always at the base of the food chain, and so we're going to talk about both of these processes. Photosynthesis is where we build a sugar. Okay, so we take CO2 and water using sun energy, and in the presence of oxygen, we build a sugar like glucose. All right. Now, if you're a plant cell and you've just made this glucose, you might need that glucose for energy storage. Maybe you don't need the energy right away, or I meant the ATP right away, and instead you want to store that glucose. Maybe you want to store it in starch. That's how plants would store. Glycogen there would be animals. I'm not sure why they have it up here next to these plants. Not so much glycogen made in plants, mostly starch, okay? On the other hand, so it could be shuttled off once it's made and stored. And through condensation reactions, starch molecules might be built. Also, this glucose could be used in condensation reactions maybe to make a cell wall, a structural component. They're not showing that here, but it is possible. In addition to that, the glucose might be used by plant cells also in cellular respiration. Yes, plants carry out respiration. They're doing that in all cells all the time. They make the glucose and then turn around and break that glucose down to make ATP. I know it seems silly, but yes, they're doing that, okay? In addition to that, animals, right? They're going to be using cellular respiration. Animals can't photosynthesize. They can't build their own glucose, but they either eat the plants or they eat an animal that has ultimately eaten plants or algae. And in that way, we ingest the glucose, right? We could also take that glucose, animals like humans, for example, could also take that glucose and build glycogen for storage. Or, as these guys are doing, probably rowing their boat here, they're going to be carrying out cellular respiration and probably fermentation as well. We do have the ability to carry out both aerobic, in the presence of oxygen, breakdown of glucose to produce lots of ATP and CO2 and water. But we also have anaerobic processes going on in our muscle cells as well, especially if we're doing those anaerobic type exercises, like rowing would be a pretty tough exercise there, requiring a lot of power at once. That would be in the absence of oxygen. Glucose then is still broken down into molecules. It's pyruvate, which is then converted to lactic acid, in humans anyway. We do make ATP, not as much. We'll look at that process too. Okay. All right. Cellular respiration. This is when glucose is oxidized. Okay. It is in the presence of oxygen, but that is not why we say glucose is oxidized. We say glucose is oxidized because bonds are broken. Okay. Bonds are broken and electrons are lost. Remember, oxidation is loss. All right. And so what happens here, you see glucose C6H12O6 in the presence of oxygen becomes CO2 and water. You see that the carbon molecule here, you see the carbon? It's lost its H's. It's lost its electrons. It's oxidized. Oxygen, on the other hand, has gained H's. It has gained electrons. It is reduced. So yes, glucose is oxidized to CO2, oxygen reduced to water. All right, how is glucose oxidized? How is oxygen reduced? Yeah, it's a long series of redox reactions, okay? That's what glycolysis and pyruvate processing and citric acid cycle and actually the electron transport chain. Overall, you are going to have some different individual reactions in there, but overall, the process is a series of redox reactions where glucose is oxidized and oxygen is reduced to water. All right, let's take a look. We're going to see 4 phases. This says 4 steps. We really should say phases because each phase then has multiple steps. And these are the phases. First, you see glycolysis. This is where the 6-carbon glucose molecule is actually split into 2 3-carbon pyruvate molecules. This is several steps. I've just listed it here as one, but it is several steps. This whole pathway, when you see glycolysis, think splitting of glucose. Okay, that's what it is. 6-carbon glucose split into 2 3-carbon pyruvates. Then the 3-carbon pyruvates are going to be oxidized further to a 2-carbon acetyl-CoA molecule. So a CO2, a carbon is removed, a CO2 comes off, and what you have left is a 2-carbon acetyl-CoA molecule. Then acetyl-CoA, this 2-carbon molecule, is further oxidized to CO2 in the citric acid cycle. We're going to see these processes as these molecules are oxidized. I'm going to show you that NAD or FAD is going to be reduced, right? Because we know oxidation reduction reactions are coupled. And as this molecule or as glucose here, or excuse me, as pyruvate is oxidized, we got this oxidation happening and carbons being removed. Well, excuse me, hydrogens being removed from the carbons. Those hydrogens and those electrons have to go somewhere, and they go to NAD or to FAD. Okay, and then in the electron transport, those reduced NADHs and FADH2s, they bring those electrons and their hydrogen protons, of course, to the electron transport chain, where those things flow down the carriers in the chain, and that is a series of redox reactions as well. That electron flow down the chain is an electrical current. It's used to power the production of ATP. All right, here it is, kind of laid out phase by phase. Here's glycolysis, the first phase. This is the 6-carbon glucose. It is split to 3-carbon pyruvates, 2 of them. Each of the 3-carbon pyruvates then get one of the carbons taken off to a 2-carbon acetyl-CoA. The acetyl-CoA enters the citric acid cycle where the remaining CO2s are broken off. And then you have the phases of oxidation here. These first 3 phases. Glucose to pyruvate, pyruvate to acetyl-CoA, acetyl-CoA to CO2. That is the complete oxidation of glucose. As glucose is oxidized, you see NAD getting reduced to NADH. He's picking up the electrons, picking up the hydrogens. Here, again, NAD is reduced to NADH. He's picked up electrons and hydrogens. Here we see FAD and FADH getting in on the fun, or FAD, excuse me, and NAD and FAD now are reduced, right? The purpose of all of this oxidation is to create these reduced electron carriers, because these guys will all join, coming this way, over to the electron transport chain, where the electrons will flow, and that's where we're going to make lots of ATP. You do see, okay, hey, we've made some ATP here. We've made a little ATP and GTP here. It's not the mass amount, though, okay? You really only have a net ATP gain here of 2 ATP per glucose. You have a few more here. Not so much. This is where the massive payoff of ATP comes in. All right, now, what does the pathway kind of look like? What can feed into it? Because yes, we're looking at glucose feeding in, but your cells can also use fats and proteins. All right, they can all feed in and act as a substrate for cellular respiration. Oh, this looks like it's a complicated pathway, but let me just show you. It is in the middle here, what we've been looking at. Here's phase 1, glucose to pyruvate, so that's glycolysis. Here's pyruvate processing to acetyl-CoA. Here's acetyl-CoA entering into this TCA, or citric acid cycle. So that is phase 1, 2, and 3, where glucose is oxidized. Up top, they're showing you how other things could feed in at different points of the pathway. At bottom, we're showing you how things could be shuttled out to build these molecules listed at the bottom of the pathway. So at the top, it's showing you catabolic, carbohydrates other than glucose. Maybe this is starch, right? Starch, a long chain of alpha glucose. And first, the alpha glucose is broken down into the sugar monomers, and then those sugar monomers are fed in as glucose and broken down. Or maybe you've eaten a steak. Maybe you've eaten some beef, I don't know, and it's a ribeye steak with lots of fats and proteins, right? That can also feed into cellular respiration. Your body can use it to generate ATP. These fats and phospholipids will not feed in at the beginning in glycolysis. They'll feed in here. The fats and phospholipids can be broken down to their glycerol components and fatty acid components. Glycerol can feed in at the midpoint of glycolysis and broken into pyruvate. The fatty acid components, on the other hand, will be broken first down to acetyl groups, and then those guys feed in here and into the citric acid cycle. The protein component of your steak, the various amino acids, depending on what they are, some are converted to pyruvate, others converted to components like acetyl-CoA, or other various components that can feed into the citric acid cycle. So, carbohydrates, fats and phospholipids, and proteins all can feed in and fuel ATP production. Also, at any point in this pathway, any of these molecules could act as an intermediate precursor to build something. Maybe your body needs to make, that pentose, that deoxyribose or ribose sugar for RNA and DNA. But you've only got glucose. So it'll start breaking the glucose and then shuttle off an intermediate molecule and convert it to that ribose or that deoxyribose, which is a 5-carbon sugar. Or maybe you've eaten lots of donuts and then you're going to go lay down. And your body's like, you know what? I've stored some glycogen. I don't need much ATP. Let's take the glucose down here. Let's convert it to phospholipids for cell membranes and fats for storage of energy. Aha! It explains how you can get fat or gain fat from eating sugars. It is possible. All right. Now, where does any of this happen? Glycolysis is in the cytoplasm. This is where glucose is split into 2 molecules of pyruvate. Let's look at it. Here is an overview schematic. This does not come from your textbook. I found this from another source. And I don't want you worrying about individual steps. What I really love about this, though, is that they are showing the individual carbon atoms here. And you see that the 6-carbon glucose is split into 2 3-carbon molecules that end up being pyruvate. Okay? As we go, you see that at first it costs the cell a couple of ATPs. There's this activation step. We add a phosphate and we add some energy to the molecule to activate it. Then we can break the phosphates back off later for payoff, right? And we can make 2 ATPs, well, a total of 4. We make 2 ATPs twice for each of the 3-carbon molecules. So you do have a net gain. We spent 2, we've gained 4 now, we have a net gain of 2. You also see a couple of reduced NADHs here being made. Yeah, there they are. Okay, so there's our gains from this. Then, well, excuse me, I should say ATP made in this pathway is made by a process called substrate level phosphorylation. This is when ATP is produced by an enzyme. All right, ADP and the phosphate bind in an enzyme's active site. They act as substrate for the enzyme. The enzyme in the active site builds the ATP and releases the ATP. Excuse me. This is how ATP is produced in glycolysis and citric acid cycle. This is not how ATP is produced in the electron transport chain. In the electron transport chain, this is called oxidative phosphorylation. This is another way to make ATP. It involves a proton gradient and a membrane protein called ATP synthase. I will show you this process. There's a few steps involved. We'll see it at the very, very end. All right, here's substrate level phosphorylation. Here's our enzyme. There's the pocket or groove that we're calling the active site. And you see ADP, the phosphorylated substrate, like maybe one of those activated 3-carbon molecules. The phosphate then is transferred off and over to produce the ATP. The ATP over here is then released, and the substrate now is also released. All right. Now, typically, glycolysis works in a pathway that is subject to feedback inhibition. This is when an enzyme in a pathway is inhibited by the actual end product of the pathway. And it's usually an early enzyme. One of the enzymes in like step 1 or step 2 is inhibited by the end product of the pathway. Glycolysis, for example, this thing is shut off when ATP is abundant. Okay, this allows cells to stop breaking down glucose and making ATP, right? If we already have enough. And then we can break it down when ATP is scarce, right? We don't want to build ATP if it's abundant. We only want to build it when it's scarce. All right. Once the pyruvate is made from glycolysis, then we move it into the mitochondria. This is where you get pyruvate processing, citric acid cycle, and the electron transport chain. These last 3 phases occur in the mitochondria. Here is acetyl-CoA. This is our 2-carbon molecule. And here is our lovely citric acid cycle, or TCA cycle, or Krebs cycle. They're really all the same thing. The reason it's called citric acid cycle is that the first product in the pathway is citric acid, or citrate. So this acetyl-CoA, this 2-carbon molecule, is first added to 4-carbon oxaloacetate to make 6-carbon citrate. Then the 2 carbons are broken off of citrate to regenerate, in the final steps, the 4-carbon oxaloacetate. So we can add another acetyl-CoA, make another 6-carbon citrate, break the 2 carbons off, regenerate the 4-carbon oxaloacetate, and continue the cycle. Add another, so on and so forth. Okay? So this is how we remove the final 2 carbons, right? And we also, along the way, generate some reduced NADH. And as we regenerate the 4-carbon oxaloacetate, we generate some FADH, more NADH. We also do make an ATP by substrate level phosphorylation, and we make a GTP, equal to ATP in energy level, well, roughly. And yes, it is also produced by substrate level phosphorylation. ATP or GTP made in glycolysis or citric acid is substrate level. All right, we're done with glucose oxidation. One thing I should tell you, I don't want you memorizing where ATP or FADH or GTP is made, any of the names of these guys. I did tell you about citrate just because I thought it was interesting since it is the name of the pathway, but please don't try to remember that. I won't ask you that. And again, this pathway, the citric acid cycle, is turned off at multiple points. It is subject to feedback inhibition. And it will basically turn on when energy supplies are low. And when energy is high, the pathway turns down or shuts itself off. These are the places where regulation can occur, some of the places anyway. You can learn a lot more about this in biochemistry later. But this first step, if there's lots of ATP, that enzyme is inhibited by ATP. Also, NADH and ATP will inhibit step 3 as well as step 4. So 1, 3, and 4 are all inhibited by high levels of potential energy in ATP and NADH-rich molecules. All right, so here is what we have summarized or sort of looked at so far. Right? We've got glucose broken into 2 pyruvates, reduced now, or oxidized, further to 2 acetyl-CoAs, and then finally completely oxidized here in citric acid cycle. You can keep up with the carbons all along the way. As we do that, we do make some ATP by substrate level phosphorylation. One of those was actually a GTP. They're lying there. Or maybe they're just not counting the GTP. And in addition to that, though, the important thing that you've really done by oxidizing the carbon molecule is making these reduced electron carriers, because they are going to go now to the final phase, the electron transport chain and oxidative phosphorylation. Yeah, this is the payoff. This is where the high potential energy of the electrons now, because glucose was oxidized, now NADH and FADH2, they contain the potential energy. It's kind of been passed on like a hot potato to these guys, right? And so now we got to harvest it from NADH and FADH in order to make the ATP. What you're going to see is a series of redox reactions through proteins that are bound in the membrane of the mitochondria in order, side by side, in what we are going to call the electron transport chain. At the end of the chain, you're going to see oxygen sitting there drawing the electrons towards it. Oxygen is very electronegative. It's like not really, but you could think of it sort of as an electron vacuum. And as the electrons pass to oxygen, oxygen is converted and reduced to water. All right, so now, these proteins in the chain, these carrier proteins, they're pretty cool. All but one of them are actually embedded in the membrane. They, ubiquinone, this guy is lipid soluble and hydrophobic and kind of moves around. I'll show you him. But the others are embedded, and they are metal-containing proteins. They have like iron and zinc and things like that that can help them actually conduct the electrical flow, the current, the electron movement. Here they kind of are. Here are the members of the chain. We're going to call them complex 1, 2, 3, and 4. And then you see Q here. He can actually move he can dock right here at complex 1, move across complex 2, and over to 3. He kind of assists. He's a proton pump, and he assists the electron moving from complex 1 across 2 and over to 3, okay? So here's what happens. NADH will come in. NADH can then give up its electron and go back to its oxidized state, NAD+. And so the electron goes this way and flows towards water, or towards oxygen to make water. The hydrogen proton itself gets pumped. And here, and even here. These are all pumps. All right, so the electron's going this way, establishing the current. And the current is used to actually power the pumping of the hydrogen ions. All right, FADH can also bring its electrons and its protons to the chain. It just doesn't go to complex 1. Instead, it brings them over to complex 2. The electron goes here to Q, back across this way, and again to water, to oxygen to make water. And the hydrogen ion in this case now needs to get pumped by Q. Okay. There's hydrogen ions really running all around in here because they don't always get grabbed by Q. Sometimes they're grabbed over here by complex 4. But you get the idea. We're getting electrons flowing. More electrons flowing. This is an electrical current. And it powers the pumping of the hydrogen ions. All right. Why do we want to pump the hydrogen ions? Because that is the goal. we flow the electrons, we establish that current, we move those electrons so that we've got this current, and then we use that current to pump protons. Why? This represents potential energy in what we call the proton motive force. All right? Once we concentrate these protons in an area inside that membrane, we can use that then as potential energy. This is kind of like water behind the dam. Okay? All right, let me show you. These guys can actually flow down their concentration gradient in a process called chemiosmosis, and that process is how we make ATP. All right, let's look. Yeah, and it's the real job of the electron transport chain. Flow the electrons, generate the current so that we can power the pumping of the hydrogen ions. We can establish the proton motive force, the gradient, and then they can flow down their gradient, yes, through an enzyme called ATP synthase, where we build the ATP. All right, ATP synthase actually acts as like a molecular motor. He is a pore protein. He's got a hole through it so that the hydrogen ions can flow. And when the hydrogen ions flow through, it causes turning. They actually, the unit spins, the F0 unit spins. Excuse me. The F0 unit spins the rotor, and as the rotor spins, the F1 unit spins. It's kind of like this spins, and that causes this to spin. They're changing shape, and this shape change allows ATP production by what we are going to call oxidative phosphorylation. Here is the giant, well, here's the process. So this is showing you these hydrogen ions in this concentrated region, all right? This is our proton motive force. Why do these guys represent potential energy? Because they do. Think about this like water trapped upstream, right? Water wants to flow downstream, and if it's held at the top of the stream or at the top of a waterfall, for example, it represents potential energy. These guys also want to flow. They don't want to flow downstream, but what they want to do is flow down their concentration gradient. All of these are like charges. They repel each other. They don't like being in this concentrated state. They really want to repulse and flow away from each other as far as they can. And so when we force them in this concentrated area, we are establishing potential energy due to their location. All right, we can open a channel. That's what this is ATP synthase. Here he is. He's a multiple. He's got quaternary structure. He's got many, many protein units. Here is the channel where these hydrogen ions can flow down their gradient. As they flow down, they are inducing chemistry changes within the channel that causes shape changes of the F0 unit, causing it to move. This causes the rotor shaft to spin, and then the F1 unit to spin and change shape. The spinning and shape changing is what allows the actual catalysis of the reaction, the building of ATP. All right, think about this like a hydroelectric dam. Let's go to that. I want to show you something. This is, for example, a hydroelectric dam where water is being held upstream. We're taking advantage now of the fact that kinetic energy, water flows downstream, so we've built this dam. We're going to stop the water behind the dam. Here's an image showing you sort of what this water would look like behind the dam. This water definitely wants to flow downstream, but we can control that, right? We can shut the dam and keep that water trapped. And when it's trapped, it's potential energy. We can open the little water flow valve here and allow the water now, this potential energy, to become kinetic as the water flows downstream. This kinetic energy of the water flowing down is used to turn a turbine. So kinetic energy of flowing water, kinetic energy of the turning turbine. This turbine is connected to a generator, which then generates electricity, which is often connected to power lines and sent off, right? Oh boy, and I just clicked that. I didn't mean to do that. Yeah, so you can see, let's see, here's a good picture. Well, maybe. I guess if you've ever seen Norris Dam, I should have pulled that one up. If you've seen that, you can clearly see like the transformers and the power lines connected. So yeah, that's what this is. This is the water behind the dam. This is the water flowing downstream. This is the turbine. Here's our transformer or generator. And here's the molecule. And I mean, of course, I'm making an analogy here. It's not water, it's hydrogen ions. And this isn't a turbine. It's the rotor shaft of ATP synthase. And we're not generating electricity, we're generating ATP. But the point is that what the cell is doing is very similar to what the dam is doing. Transformations, potential to kinetic to now electrical energy. The cell is doing potential to kinetic to potential energy. It's in energy transformations here. All right, let's go back. All right, this is the vast majority of the payoff. This is where the money comes. Your book says that per glucose, 29 ATP molecules are made. I have seen some books that'll say something like 32 to 34, and it really does depend on the exact cell type and the conditions, you know, how much oxygen's available and the exact conditions of the cell. And so I really don't want you to focus on the total amount, but I mean, it is somewhere between 29 and 32 ATPs about. And what I want you to know is that the majority of them come here in this final phase, right? Only 4 are made in glycolysis and citric acid cycle during cell respiration. Yeah, here's the total thing. The total summary of cellular respiration: phase 1, phase 2, phase 3, and now it's tied to phase 4 and oxidative phosphorylation. All right, beautiful. Now, what I'm showing you here, obviously, is oxygen sitting at the end of the electron transport chain, acting as sort of the final acceptor, the final resting place, if you will, of the electrons and the protons in water, right? And the water's just wicked away. But what happens if you're a prokaryote? Because I mean, eukaryotes really. Okay, so we've got, if we're doing respiration, we're doing aerobic respiration. But some prokaryotes can actually carry out anaerobic respiration, and they can use an alternative electron acceptor. They could actually, you know, have glycolysis and pyruvate processing and citric acid and an electron transport chain. They would have all that, but instead of oxygen sitting here, maybe it's nitrate, or maybe it's sulfate, or some kind of phosphate. Like if you're E. coli, you could do that. You can use oxygen if it's available, but if it's not available, well, what about nitrate? Is it around? Yep, it is. We'll use it. Or if nitrate's not around, what about sulfate? Yeah, they've got a variety that they can use. It's really cool. We say this is anaerobic respiration if it is all of these phases, including the electron transport chain, but the molecule here is not oxygen. It is something other than oxygen. That's anaerobic respiration, okay? But what if no final acceptor's not around? What if you're a eukaryote and oxygen's absent? Or if you're a prokaryote and oxygen's absent, but also sulfates and nitrates are absent? None of them around. In that case, if there's no electron acceptor, this is when fermentation becomes the option. Fermentation is going to be a lot less efficient as far as ATP production goes, because you're only going to generate 2 ATP per glucose, not the 29 that you would get here. So you're going to do 2 ATP per glucose. So not very beneficial as far as ATP numbers go. However, it is beneficial if it allows you to survive in an anoxic environment. Or it is beneficial to allow you to keep exercising, especially in exercises that would require so much power and such a burst of ATP that there's no way you could deliver the oxygen in time. In the right quantities. That's when fermentation comes in. So yeah, your book does kind of say, okay, oxygen's the most effective, it's highly electronegative, there's a huge difference between the potential energy of NADH and O2, and that difference when the electrons are transferred, that reaction, you get a huge release of energy. It allows the generation of a really nice proton motive force. And cells that don't use oxygen as an electron acceptor cannot generate a large potential energy, and they make less ATP. Fermentation, even less. Let me make sure that I did not skip over something. Yeah, fermentation is the only option in eukaryotes if oxygen's absent, because they can't carry out anaerobic. We can't use nitrates or sulfates. It'd be cool if we could, but we can't. So yeah, in fermentation, this is glycolysis, basically. And that's all it is. Lactic acid fermentation, what happens is glucose is broken to pyruvate, and then the pyruvate is converted to lactic acid. In alcoholic fermentation, glucose is broken to pyruvate, and the pyruvate is then converted to ethanol as waste, right? Here's sort of the process. Glucose to pyruvate. You make 2 ATPs. That's all the energy we've got. We do generate 2 NADHs, but we can't use them because we don't have an electron transport chain. So what happens is this pyruvate is converted to lactic acid just so we can regenerate the NAD so that glycolysis can continue, because NAD is required for glycolysis to happen to make the 2 ATP. In alcoholic fermentation, glucose again is oxidized to pyruvate, broken down to pyruvate. We get 2 ATP, and we do generate 2 NADHs, but again, we can't use them. We don't have an electron transport chain. So in this case, they don't have the same enzymes, and so they convert the pyruvate first to this and then to ethanol. And the purpose of that is to regenerate the NAD so that glycolysis can continue and we can still make 2 ATP per glucose. So we're really only making 2 ATP per glucose, and we're carrying out this final step just to regenerate the NAD so we can continue to make 2 ATP per glucose. Yeah, so not very big payoff there. However, it is a huge benefit. Like I said, in yeast and other prokaryotes, they can survive. They can survive in environments with no electron acceptor. It's very good for survival rates. And in humans, it's pretty good to generate a huge burst of ATP quickly and without the need to deliver oxygen. We do this in our anaerobic exercises, like if you're weightlifting or doing any kind of anaerobic type exercise. This is what's happening. Now, the lactic acid in humans, in yeast, actually, the waste is really kind of just released, and it can actually build up to the point that it kills the yeast because ethanol is toxic. Lactic acid is also toxic. Some people say it's what causes muscles to burn. Other people are like, no, it's tiny microfibers. And they say lactic acid doesn't really remain around because your body would take lactic acid, convert it back to pyruvate once your anaerobic exercise or anaerobic demand is over. The lactic acid would be converted back to pyruvate, and the pyruvate then would be processed and enter into the citric acid cycle to be continued to break down. Your body is not going to waste that. There's still a lot of energy in it. So it can be used, it can be produced just to generate a huge burst of ATP, but then it's not going to just build up in the muscles. It's going to be converted back and then broken down further by cell respiration to get the max energy payoff. Yep, so that's sort of the process. Fermentation, of course, yeah, compared with respiration, extremely inefficient, but it does have its benefits. Your book says organisms never use fermentation if an appropriate electron acceptor is available. This is absolutely false. It's absolutely false. They definitely use fermentation in the case of anaerobic type exercises. Multicelled organisms do. Also, something that's really interesting, they have shown that cells that are in a tumor, like cancer cells, they carry out fermentation even in the presence of oxygen, which is very interesting. People think that they're carrying out this fermentation so that they can then take the lactic acid and use it as a precursor to then build components, because these tumor cells are growing and reproducing proliferatively. And so they need lots of molecules as substrates to build like all of their cell membranes and build their carbohydrates and build all their components. So they think that since cancer cells also have lots of blood flow coming to them. They're getting a rich supply of glucose, so they're not going to be scarce of glucose. They can afford to break down glucose and make only 2 ATP because they get tons of glucose, and then they're taking this molecule and using it as building starting points, precursors. So yeah, some organisms and some cells do use fermentation. This is a flat-out lie in your textbook. All right, so that is the process of cell respiration. We will talk about this again when we come back from fall break. Now what I want you all to do, I want you all to go to our Canvas site. I want you to click on My Lab and Mastering. Let me just show you real quick. I want you to open it up. And if this isn't how you get to Learning Catalytics, you can get to Learning Catalytics however you usually do. This is how I get there every time, so I'm sure I could do it easier, but I don't. And what you're going to look for is a module. Let's see, where is it? Here it is, Cellular Respiration. And I know you won't have this many modules. You won't have this one that says instructor-led. You should just have one that says Cell Respiration, Self-Test. It's got 5 questions. I have set this thing available from Sunday, September 30th at noon, all the way until Sunday, October 7th at noon. So at any point, you can, between that time frame, click on this, log in, answer these Learning Catalytic questions based on that presentation that you just watched for some points. And I hope you all have a good rest of fall break, and I'll see y'all soon.